Welcome to Chapter 10, Biotechnology. In this chapter, we're going to go over the following sections: a little discussion of cloning and genetic engineering, followed by biotechnology in medicine and agriculture, and then some information on genomics and proteomics. Pictured here is a thermal cycler on the left and the hot spring of Yellowstone National Park, which is an example of one of the places where the bacteria came from that enables us to use this thermal cycler in the process called PCR, which will be under discussion here shortly. Section 1, Cloning and Genetic Engineering. By the end of this section, you will be able to explain the basic techniques used to manipulate genetic material, explain molecular and reproductive cloning. The latter half of the 20th century began with the discovery of the structure of DNA, then progressed to the development of the basic tools used to study and manipulate DNA. These advances, as well as advances in our understanding of and ability to manipulate cells, have led some to refer to the 21st century as the biotechnology century. The rate of discovery and of the development of new applications in medicine, agriculture, and energy is expected to accelerate, bringing huge benefits to humankind and perhaps also significant risks. Many of these developments are expected to raise significant ethical and social questions that human societies have not yet had to consider. Biotechnology is the use of artificial methods to modify the genetic material of living organisms or cells to produce novel compounds or to perform new functions. Biotechnology has been used for improving livestock and crops since the beginning of agriculture through selective breeding. Since the discovery of the structure of DNA in 1953, and particularly since the development of tools and methods to manipulate DNA in the 1970s, biotechnology has become synonymous with the manipulation of organisms' DNA at the molecular level. The primary applications of this technology are in medicine, for the production of vaccines and antibiotics, and in agriculture, for the genetic modification of crops. Biotechnology also has many industrial applications, such as fermentation, the treatment of oil spills, and the production of biofuels, as well as many household applications, such as the use of enzymes in laundry detergent. Manipulating genetic material. To accomplish the applications described above, biotechnologists must be able to extract, manipulate, and analyze nucleic acids. Review of nucleic acid structure. To understand the basic techniques used to work with nucleic acids, remember that nucleic acids are macromolecules made of nucleotides, which consist of a sugar, a phosphate, and a nitrogenous base. The phosphate groups on these molecules each have a net negative charge. An entire set of DNA molecules in the nucleus of eukaryotic organisms is called the genome. DNA has two complementary strands linked by hydrogen bonds between the paired bases. Unlike DNA in eukaryotic cells, RNA molecules leave the nucleus. Messenger RNA, mRNA, is analyzed most frequently because it represents the protein coding genes that are being expressed in the cell. Isolation of nucleic acids. To study or manipulate nucleic acids, the DNA must first be extracted from cells. Various techniques are used to extract different types of DNA. And this figure illustrates one method, a very common method. Most nucleic acid extraction techniques involve steps to break open the cell and then the use of enzymatic reactions to destroy all undesired macromolecules. Cells are broken open using a detergent solution containing buffering compounds. To prevent degradation and contamination, macromolecules such as proteins and RNA are inactivated using enzymes. The DNA is then brought out of solution using alcohol. So it precipitates into something solid, which by this picture is being depicted as being attached to the glass rod here. This is most common in demonstrations. I've never actually done this in the lab when I've done a DNA extraction. I'm generally using chemicals to precipitate out the DNA, and then I can take that and put it back into solution, and I have additional purifications that are available as well. The resulting DNA because it is made up of long polymers, forms a gelatinous mass in this protocol depicted here. RNA is studied to understand gene expression patterns in cells. RNA is naturally very unstable because enzymes that break down RNA are commonly present in nature. Some are even secreted by our own skin and are very difficult to inactivate. Similar to DNA extraction, RNA extraction involves the use of various buffers and enzymes to inactivate other macromolecules and preserve only the RNA. As you can imagine, the steps used to preserve RNA are more extensive than that for DNA. Gel electrophoresis. Because nucleic acids are negatively charged ions at neutral or alkaline pH in an aqueous environment, they can be moved by an electric field. Gel electrophoresis is a technique used to separate charged molecules on the basis of size and charge. The nucleic acids can be separated as whole chromosomes or as fragments. The nucleic acids are loaded into a slot at one end of a gel matrix. An electric current is applied, and negatively charged molecules are pulled towards the opposite end of the gel, the end with the positive electrode. Smaller molecules move through the pores in the gel faster than larger molecules. This difference in the rate of migration separates the fragments on the basis of size. The nucleic acids in a gel matrix are invisible until they are stained with a compound that allows them to be seen, such as a dye. Distinct fragments of nucleic acids appear as bands at specific distances from the top of the gel, the negative electrode end, that are based on their size. A mixture of many fragments of varying sizes appear as a long smear, whereas uncut genomic DNA is usually too large to run through a gel and forms a single large band at the top of a gel. So pictured here is an example of a gel. We have the lanes at the top. The first lane and the last lane has a size marker in it. And this is typically what you do. At least one size marker lane. And then in here you see the smallest fragments have accrued down here. Then you have some larger, more clear fragments. Usually what gets to the bottom the fastest is stuff you're not looking for if you're using a gel. Anyway, but these, this might be like 1.5 kilobase, this is about, or 1 kilobase, I'm not sure what ladder this is. 10 kilobase ladders are pretty common, so that's 10,000 base pairs. And then in each of these lanes through here, you know, this lane number 2 and 3 has at least one fragment that's the same size, so this might be the result of a digestion or something, but I have tons and tons of gel pictures, and okay, I can't go on about it all day. Okay, so polymerase chain reaction. DNA analysis often requires focusing on one or more specific regions of the genome. It also frequently involves situations in which only one or a few copies of DNA molecule are available for further analysis. These amounts are insufficient for most procedures, such as gel electrophoresis. If you really only had a single copy of a DNA molecule to run through that gel, you would not see it. Polymerase chain reaction, PCR, is a technique used to rapidly increase the number of copies of specific regions of DNA for further analyses. And that's what we are seeing in this figure. PCR uses a special form of DNA polymerase. Remember, that's the molecule that carries out the replication of DNA, as in the last chapter, and other short nucleotide sequences called primers that base pair to a specific portion of the DNA being replicated. PCR is used for many purposes in laboratories. These include the identification of the owner of a DNA sample left at a crime scene, paternity analysis, 3, the comparison of small amounts of ancient DNA with modern organisms, and 4, determining the sequence of nucleotides in a specific region. So I'm not playing that video for this reading of the textbook, but if you look up the PCR song, there's a song that will walk you through this process. and it's pretty funny. So this process, the book doesn't go on about it much, but I'm going to talk a little bit about it. You have the double-stranded DNA. First, you have to melt that. You have to separate it out into single-stranded components. Then you'll have two primers. These primers will bracket the gene of interest, or the area that you want to make copies of. So you have two primers that will anneal, and as you know, you can add nucleotides to the 3' end with your DNA polymerase. So you also need a bunch of dNTPs available. You have to have a pool of those to be added. And so you have DNA synthesis will occur in both directions in a continuous fashion. And then that's the extension step. So when you anneal the primers in the first place, the second step is annealing, and this is generally at a lower temperature. You design these primers, and they're in sufficient quantities that you favor the binding of the primer to your target DNA over the target DNA simply re-annealing. So denaturing, annealing, and then extension with DNA polymerase. Now after that, you repeat that cycle many times, usually 35 to 40 times if you're talking about standard PCR. Denaturing, annealing, and extension. And so you need a thermal cycler to run that on. You need your DNA template, buffer, primers, dNTPs, and of course DNA polymerase, which is quite expensive material for the lab. So that's a very quick lesson on PCR. Cloning. In general, cloning means the creation of a perfect replica. Typically, the word is used to describe the creation of a genetically identical copy. In biology, the recreation of a whole organism is referred to as reproductive cloning. Long before attempts were made to clone an entire organism, researchers learned how to copy short stretches of DNA, a process that is referred to as molecular cloning. Molecular cloning. Cloning allows for the creation of multiple copies of genes, expression of genes, and the study of specific genes. To get the DNA fragment into a bacterial cell in a form that will be copied or expressed, the fragment is first inserted into a plasmid. A plasmid, also called a vector in this context, is a small circular DNA molecule that replicates independently of the chromosomal DNA in bacteria. In cloning, the plasmid molecules can be used to provide a vehicle in which to insert a desired DNA fragment. Modified plasmids are usually reintroduced into a bacterial host for replication. As the bacteria divide, they copy their own DNA, including the plasmids. The inserted DNA fragment is copied along with the rest of the bacterial DNA. In a bacterial cell, the fragment of DNA from the human genome, or another organism that is being studied, is referred to as foreign DNA to differentiate it from the DNA of the bacterium, which is the host DNA. And in this picture here, your insert, your foreign DNA, would go into right here into the multiple cloning site. So this is beyond the scope of this textbook, but I just wanted to share this with you. Plasmids occur naturally in bacterial populations, such as Escherichia coli, which is where this plasmid series was initially discovered, and have genes that can contribute favorable traits to the organism, such as antibiotic resistance, the ability to be unaffected by antibiotics. And in here, we're looking at this AMP is for ampicillin resistance, which is a beta-lactamase gene. Plasmids have been highly engineered as vectors for molecular cloning and for subsequent large-scale production of important molecules, such as insulin. A valuable characteristic of plasmid vectors is the ease with which foreign DNA fragments can be introduced. These plasmid vectors contain many short DNA sequences that can be cut with different commonly available restriction enzymes. That's what all these are. These are different restriction enzymes. Restriction enzymes, also called restriction endonucleases, and they're endonucleases because they cut inside of a sequence, recognize specific DNA sequences and cut them in a predictable manner. They are naturally produced by bacteria as defense mechanisms against foreign DNA, most commonly introduced by bacteriophages, by the viruses that use bacteria cells as hosts. Many restriction enzymes make staggered cuts in the two strands of DNA, such that the cut ends have a 2 to 4 nucleotide single-stranded overhang. That's what's depicted here. This is the overhang. We also call them sticky ends. Wait, did I already say that? No, I didn't. Okay. The sequence that is recognized by the restriction enzyme is a 4 to 8 nucleotide sequence that is a palindrome. And that is for the restriction enzyme class that we're talking about here. There's other restriction enzymes that have different types of recognition sites, but yeah. Like with a word palindrome, this means that the sequence reads the same forwards and backwards. In most cases, the sequence reads the same forward on one strand and backwards on the complementary strand. When a staggered cut is made in a sequence like this, the overhangs are complementary. So these two sticky ends, if you let them, they could re-anneal. They could reconnect here and base pair. Then you would need another enzyme to fix the backbone. But usually you don't want to just do that. You want to use these sticky ends to stick this piece into another piece of DNA that will have a complementary sticky end. Because these overhangs are capable of coming back together by hydrogen bonding with complementary overhangs on a piece of DNA cut with the same restriction enzyme, these are called sticky ends. Okay, sorry I spoiled that for you ahead of time. The process of forming hydrogen bonds between complementary sequences on single strands to form double-stranded DNA is called annealing. Addition of an enzyme called DNA ligase, which takes part in DNA replication in cells, permanently joins the DNA fragments when the sticky ends come together. In this way, any DNA fragment can be spliced between two ends of a plasmid DNA that has been cut with the same restriction enzyme. Extra aside, New England Biolabs is the vendor, and I'm not being paid by them at all, but they are a very popular vendor of restriction endonucleases. And they have, here's a little poster that came from them. They have 285 restriction enzymes available that are able to use a single buffer now. So each restriction endonuclease could have a slightly different buffering condition under which it operates most efficiently, but NEB has configured these so that they have a single buffer system that works really well. So I'm going to talk through this figure, and this is the figure talking about molecular cloning in a little bit more detail. So we have our foreign DNA, here's our plasmid. We use the restriction enzymes to make the sticky ends, and then these can anneal, and then DNA ligase can seal. So the LAC-Z gene here is being used as a reporter, in that if the insert doesn't happen, this reporter gene will work as it's supposed to, thereby giving you a color when it reacts with another chemical in when you plate your transformants, which is downstream of this. Okay. So we take these plasmids that we put hopefully our foreign DNA, our insert is another way to call it. Hopefully our insert is in our plasmid, and then we take these cells that we've made competent. Mostly we do this, well, chemical competent protocols are most common. You can also use electroporation. You can use electricity. So what we need to do, most cells don't naturally take up naked DNA. It doesn't make a lot of sense to just take up any old DNA that's in your environment. But if they do, they'll have a DNA ACE, an enzyme to cut it up to basically eat it. Here, we need to make this bacteria more willing to take up naked DNA, so we'll use either chemicals to basically punch some holes in the cells without killing the cell, or use electricity to accomplish the same thing. Electricity is really good at it, by the way. Electrocompetent cells are much more efficient at taking up naked DNA. So if you're planning a reaction, you need to factor that into how much you're going to use when you plate it. Okay, so here, so the bacteria that take up the recombinant plasmid cannot make the enzyme from the gene that the fragment was inserted into, which I mentioned, LAC-Z. Now on the plate that you put it on, you'll have a chemical that will change color when LAC-Z acts upon it. And so these are ones that will turn blue. So any white colonies that grow, number one, it had to get the plasmid because it's resistant to ampicillin, because you put ampicillin on this plate as well to screen out any bacteria that did not get a plasmid at all. And then number two, the ones that have the insert where you want it will produce colonies that are white. And then finally, there's some troubleshooting you might have to do sometimes if your insert is really small, or if it happens to be in a length that is divisible exactly by 3, you can sometimes have an odd cloning, a molecular cloning effect where you achieve blue colonies throughout, even though some of them are transformants and they do have your insert on the plasmid. The LAC-Z reporter gene might still be active. This is probably the very, very small number of these actually happen. I've sometimes pinned my hopes on that just maybe for whatever reason the LAC-Z was still functional, because I just hoped that my transformation wasn't a complete bust. Because these take a little while to set up, and you want it to work out well for you. But if you can't find the insert, so you'll take your transformants, the ones you think are transformants, you'll grow them up in great density. Then you can extract their DNA. You can use restriction enzymes to look for that fragment, that insert that should be there. And so generally, well, for me, that's almost always been in vain. I've never had like a light blue colony that turned out to actually be my transformant of interest. Yeah. Anyway, there's a little segue into a little more detail in the molecular cloning world. If you go into any biotechnology, this molecular cloning is super standard and very common kind of protocol to do. Now, reproductive cloning, which is what most people probably think of when they hear the term clone. You think, was it sixth day or something like that. So reproductive cloning is a method used to make a clone or an identical copy of an entire multicellular organism. Most multicellular organisms undergo reproduction by sexual means, which involves the contribution of DNA from 2 individuals, the parents, making it impossible to generate an identical copy or a clone of either parent. Recent advances in biotechnology have made it possible to reproductively clone mammals in the laboratory. Natural sexual reproduction involves the union during fertilization of a sperm and an egg. Each of these gametes is haploid, meaning they contain one set of chromosomes in their nuclei. The resulting cell, or zygote, is then diploid and contains 2 sets of chromosomes. This cell divides mitotically to produce a multicellular organism. However, the union of just any 2 cells cannot produce a viable zygote. There are components in the cytoplasm of an egg cell that are essential for the early development of the embryo during its first few cell divisions. Without these provisions, there would be no subsequent development. Therefore, to produce a new individual, both a diploid genetic complement and an egg cytoplasm are required. The approach to produce an artificially cloned individual is to take the egg cell of one individual and to remove the haploid nucleus. Then a diploid nucleus from a body cell of the second individual, the donor, is put into the egg cell. The egg is then stimulated to divide so that development proceeds. This sounds simple, but in fact it takes many attempts before each of the steps is completed successfully. So here, enucleation is what that's called when you remove the haploid nucleus from the egg cell. And then this is done electrically, this pulse, to get it to fuse and to transfer that nucleus over and to get it to start growing. And then you would transplant that into a surrogate mother, which is one of the reasons why the question of human cloning has, runs up against very strong ethical concerns, right? There have been attempts at producing cloned human embryos as a source of embryonic stem cells. In the procedure, the DNA from an adult human is introduced into a human egg cell, which is then stimulated to divide. The technology is similar to the technology that was used to produce Dolly, but the embryo is never implanted into a surrogate mother. The cells produced are called embryonic stem cells because they have the capacity to develop into many different kinds of cells, such as muscle or nerve. The stem cells could be used to research and ultimately provide therapeutic applications, such as replacing damaged tissues. The benefit of cloning in this instance is that the cells used to regenerate new tissues would be a perfect match to the donor of the original DNA. For example, a leukemia patient would not require a sibling with a tissue match for a bone marrow transplant. And even then, sometimes you need immune system suppressors, immunosuppressive therapy, right? Now another, this is on my own, this isn't from the OpenStax book, but Viagen will clone your horse if you would like that. That's $85,000 to $150,000. In general, they'll clone your cat for $25,000, clone your dog for $50,000. And that's, I mean, your animals are family, right? So if you have the kind of resources, go for it. Genetic engineering. Using recombinant DNA technology to modify an organism's DNA to achieve desirable traits is called genetic engineering. Addition of foreign DNA in the form of recombinant DNA vectors that are generated by molecular cloning is the most common method of genetic engineering. An organism that receives the recombinant DNA is called a genetically modified organism, GMO. If the foreign DNA that is introduced comes from a different species, the host organism is called transgenic. Bacteria, plants, and animals have been genetically modified since the early 1970s for academic, medical, agricultural, and industrial purposes. These applications will be examined in more detail in the next module. Although the classic methods of studying the function of genes began with a given phenotype and determined the genetic basis of that phenotype, modern techniques allow researchers to start at the DNA sequence level and ask, what does this gene or DNA element do? This technique, called reverse genetics, has resulted in reversing the classical genetics methodology. One example of this method is analogous to damaging a body part to determining its function. So gene knockouts is what is being discussed here. An insect that loses a wing cannot fly, which means the wing's function is flight. The classic genetic method compares insects that cannot fly with insects that can fly and observe that the non-flying insects have lost wings. Similarly, in a reverse genetics approach, mutating or deleting genes provides researchers with clues about gene function. Alternatively, the reverse genetics can be used to cause a gene to overexpress itself to determine what phenotype effects may occur. So we have techniques to silence genes, knock them out, change their function, and then we can restore that function. And usually in these kinds of studies, that's what you have to do. You have to knock the gene out first, demonstrate the change in phenotype, and then reintroduce that gene and show that that phenotype's been restored. That's, I almost hate the term, but it seems to apply, the golden standard of a lot of genetic research. When you can do it, with gene silencing, you can introduce something that will temporarily turn off a particular gene, observe the change in function, and then when that silencing mechanism is alleviated, the function is restored. And that will help determine that that gene plays a role in that phenotype, that metabolic function, that whatever you're looking for. And that brings us to the end of section 10.1. Join me next time for section 2, Biotechnology in Medicine and Agriculture. Until then.